So we're covering the lymphatic system. Exercise 1 is a lot of identification of different components of the lymphatic system. So the open ends of these white tubes here represent lymphatic capillaries, and this lacteal here in the small intestine, is technically a capillary as well. It really functions to absorb dietary fats and transport it to the subclavian veins. But we have the lacteal here. We have these little open-ended, the white ends of these tubes, I guess you could say. Then we have our lymphatic vessels, which are the larger vessels. And they merge to form larger ducts that transport lymph fluid, once it's collected from the interstitial spaces, into the right subclavian vein and the left subclavian vein, respectively. So within that, we have the lymph nodes, which is where we filter the lymph that we've absorbed from the interstitial spaces. From the lower extremities, those will merge to form the cisterna chyli, which is best when you're looking at this model to stand at her head and look downward, because then you can see it. So either from that end there or from this end looking down this way. It's located behind the hepatic portal vein, so it's this white dilated region. And superior to that is where you had the beginning of the thoracic duct. That's why they call it the thoracic duct. So this long white tube that ultimately merges with the left subclavian vein. Lymph from the right arm, right side of the head, right chest will pass through the right lymphatic duct into the right subclavian vein. Some people will refer to this, the thoracic duct, as a left lymphatic duct, but I still use thoracic duct because that's generally what most physicians are going to use, so I would use that term. Here's your hepatic portal vein. And that's it for that model. On this model, there's additional, what's called the MALTs, the mucosa-associated lymphatic tissues. That would include things like the Peyer's patch. The Peyer's patch is found in the mucosa layer of the small intestine. And it responds to different bacteria and so forth that can get across the lining of your small intestine. So it stimulates your immune system from that point because it's picking up on microbial invasion in the small intestine. In the mouth, or in the nasopharynx, there's going to be a tonsil. That's F here on the back. That has the same kind of reactionary function as the Peyer's patch does, but it's located here at the entrance point of food and air. So it responds primarily to pathogens, that are in the air that you breathe in and out through the nose. Then we also have the appendix, which is considered part of this lab, but generally we don't even have an example out here, so I guess no one's using it. But the appendix is found here between the small intestine and the large intestine. We'll really look at that when we get to the digestive system, so it'll be found actually right about there. But I don't think anybody points to it really at this point in time. Then we have red bone marrow, which, I forgot about that one. So you have these bones here that are to represent, here we'll put it this way so it has a green background, red bone marrow. And that's where, oopsie doodle, all blood cells originate from. So this is going to be that will be the source of your specific and non-specific cell responses, in immunity. It's also where B lymphocytes will mature and stay to mature. And then we have the little naked dude on a plate. Here we have our baby model. This is the thymus gland. The thymus gland produces the hormone thymosin, and thymosin stimulates T lymphocyte maturation, or immunocompetence, it's called. It means it becomes its functional in the immune system, and that's where the T lymphocytes will mature. And let's see, I think that's most of part 1. Then we have exercise 2 is histology. So we're going to look at the lymph node and to identify the cortex versus the medullary area, and maybe just tell me what cells will be there. And then we move on to the immune response, the innate and adaptive immunity. So I think the easiest way to distinguish between those two things, so we can go over to the blood cell models over here, is to look at your white blood cells. And once you know what is part of your specific immunity, you know, which is going to be T lymphocytes and B lymphocytes, or T cells, B cells, anything else that you look at is part of that non-specific, you know, immunity. So we can come back to that if you like, but so we're going to look at the other components of sort of your non-specific first line of defense versus second line of defense. So your first line of defense is going to be skin and secretions to the skin. So now we need to find, I know this is all scattered all over because we don't have that many models, but we need, where's the skin? There's a little skin model out here.
No, that's not it. With the Pacinian corpuscles and stuff on it, right? I saw.
Yeah, yes, the ones with it. Where is it? It's a little short.
I know, it was here.
It was just here yesterday. So I just need to find it.
I don't think it's
It
here.
is now, someone moved it. So we are going to get see, because the
diagram's
key
there.
for it is out here. I don't know what happened to it. So let me get it. Which is to the superior aspect. You have, so the stratified nature of that tissue, that's a good idea. All right, the stratified nature of the epidermis in and of itself is a physical barrier. We also have dendritic cells in there that are a type of lymphocyte, so to speak, or a phagocytic type of a cell. And then remember, we have our sweat glands and our oil glands, or our sudoriferous glands and our sebaceous glands, that produce secretions on the surface of the skin that help act as an antimicrobial as well. So that's the primary function of that skin model, is to represent those particular secretions. Along the respiratory and digestive pathways, we secrete a lot of mucus, so that also is a physical barrier to bacteria, you know, viruses entering the body. And we kind of look at that again in both the respiratory and digestive systems. So we'll kind of come back to that. Then you have the defenses that are now within the body itself. So that takes us to the cells and to the different, you know, secretions, you know, or chemicals that circulate in plasma. So the cells that we want to look at now, we'll kind of come back to, are your phagocytic cells. So the two that are primarily phagocytic are going to be our neutrophil, right? That's pretty much bacteria and some fungi that it will phagocytize. And then we have our big mother monocyte that you gave me, mono. Dr. Brightman loves that. That is capable of phagocytosis of really anything. You know, so these make really good antigen-presenting cells, because they can pretty much incorporate about anything in there, take the antigen, put it on its cell surface, and become a really effective antigen-presenting cell to those T lymphocytes, you know, the helper cells. So we have our phagocytic cells. We have interferon, you know, which interferes with viral replication. We have transferrin that interacts and takes up available iron in serum or plasma to make it unavailable essentially to bacteria, so that kind of inhibits their microbial replication. And then we have our C proteins, which, you know, the complement system, which is like your C3s and primarily your C5s, that we don't have a model for that. You should just know that it's there, you know, and then that it's one of those secretions. That takes us to the specific line, which are going to be your T lymphocytes and B lymphocytes. So we know that with T lymphocytes, we have helper T cells, which are your CD4s or your T4s, and you have your cytotoxic cells that are your CD8s or T8 cells. The helper cell receives the information from the antigen-presenting cell, and then it can then stimulate a B lymphocyte to become a plasma cell and make antibodies, and also give that antigen to the T lymphocytes so they can, or the T8, so they can make the cytolytic granules it needs to make to kill that particular organism. And I think that's pretty much about it. Okay. That's our specific and non-specific. I will tell you that generally, as you know, like I try to use these larger models first, you know, for testing purposes. I don't generally use the little dudes. And if you need to distinguish, if the question requires you to distinguish between a B lymphocyte or T lymphocyte, it's going to be stated in the question. All right, so, you know, it might say, I have an A on here, and I said, if A is a T lymphocyte, you know, is it part of specific or non-specific immunity? Or I could say, what is its specific function? Okay. And so it would have to be given to you because clearly all the lymphocyte models all look the same. So out of fairness. And I think that's it.